In the previous part, we discussed that altered or unnatural bases are incorporated in the DNA under the influence of the reactive oxygen species. The DNA repair system can repair these lesions, and the modified bases or nucleotides removed by the repair process ends up getting excreted, and those can be detected in urine. This forms the premise of a study that was published in the Proceedings of the National Academy of Sciences USA in the year 1988. The details of this publication are presented in the bottom of the slide. Here we will look at the results from this study. They recruited few mice and monkeys, and they collected urine samples over a 24-hour period of time. Then they estimated thymine glycol and thymidine glycol in the urine in terms of nanomoles per kg per day. Take a note of this unit, nanomoles per kg per day. You can see that the urinary output of these analytes is normalized against their body weights. For the mice, the normalized urinary output of thymine glycol or thymidine glycol were about 6 and 2.6 nanomoles per kg per day respectively, whereas the same for monkeys were about 1 nanomole per kg per day for both the analytes. The data clearly shows that the normalized urinary output of thymine glycol and thymidine glycol is lower in monkeys compared to mice. The authors went on to collect data on urinary output of thymine glycol and thymidine glycol in various species such as mice, monkeys, rats, and humans. They also measured their oxygen consumption normalized against their body weights. Oxygen consumption rate was expressed as milliliters of oxygen per gram per hour. They then visualized the data in form of a graph. The urinary output plotted on the y-axis and oxygen consumption plotted on the x-axis. Before we look at the data, let us first understand what does oxygen consumption rate tell us. It tells us about the rate of metabolic activity that requires oxygen. Hence, higher oxygen consumption rate indicates higher metabolic rate. On the other hand, higher urinary output of modified DNA bases indicate higher frequency of DNA damage under oxidative environment. Now here is the simplified representation of the data based on the urinary output of thymine glycol. The results on the basis of urinary output of thymidine glycol is similar in trend, and one can look into their article. The details of the article are mentioned in the bottom of the slide. Look at the mice. They have higher rate of oxygen consumption and also higher urinary output of thymine glycol. Look at the humans. They have lower rate of oxygen consumption and lower urinary output of thymine glycol. Taking into account the data from all the species, a trend is clearly visible. There appears to be a direct relationship between urinary output of the modified bases and oxygen consumption rate. More the oxygen consumption rate, more is the urinary output of the modified bases. And that leads to the following interpretation. More the metabolic activity, more frequent is the attack on the DNA. If this is true, you can think what kind of effect will a compromised DNA repair system will have on the cells. The rate of oxidative damage will be high if the metabolic activity is high. One last thing before we close this part. If you consider the species indicated in the schematic, the average lifespan increases from mouse to rat to monkey to human. So how about this interpretation? That the lifespan is inversely related to the rate of metabolic activity. Strange or interesting? See you in the next part, where we will continue to discuss on the results with some more insights.